So when we're talking about spectroscopic methods, is mostly the interaction of matter with light. Okay, so it could include molecular or atomic spectroscopy. But the one that I'm going to tackle today is more on molecular spectroscopy. But the experiment that you're going to do is atomic spectroscopy. Okay, so spectroscopy, I think, is just possible because of this so-called electromagnetic spectrum. Okay, so this is a region wherein different electromagnetic radiation is observed, and usually the one that is more familiar to that in this region is this color region. But unfortunately, this is just a small what we call region in the electromagnetic spectrum. But the other electromagnetic radiation that we have, like infrared, ultraviolets, okay, we take advantage of them and use them to develop some sort of a spectroscopic method. So if you're going to look at here, you might say that ultraviolets, which is in a shorter wavelength but higher energy, okay, compared to what we call the infrared region, okay, which is a longer wavelength but longer wavelength. Okay, so it's what we call longer wavelength, but we could say lower energy. Okay, so if we're going to look at the spectroscopic methods that were developed, so we could say this is how it's tabulated, and the type of we could say quantum transition that it has. So we have the gamma ray emission. Okay, so the type of quantum transition here is in nuclear. And then we have the X-ray absorption, emission, fluorescence, and diffraction. So this is an XRF and XRD. So the usual wavelength is 0.1 to 100 Armstrong. So the shorter the wavelength, the more energetic they are. Okay, so the one that it affects is the inner electron. And then you have the absorption. So you can have the vacuum ultraviolet, which affects the bonding electrons. It's a 10 to 180 nanometer. And then you have what we know as the UVBs, okay? UV visible as absorbance or absorption. And you also include here the so-called emission and fluorescence. So this is the typical thing that we observe in the visible region. And it involves the bonding electron, okay? And then we also have this infrared absorption or Raman scattering. So it's a much longer wavelength than the absorbance and fluorescence. And they just what we call the vibrational spectroscopy. Okay, so I have experience at least in the vibrational spectroscopy and the absorbance and the fluorescence spectroscopy. And then you also have the microwave absorption, the electron spin resonance, and the nuclear magnetic resonance, the so-called NMR. And it deals with the rotation of the molecules, the spin of the electrons in the magnetic field, and for the NMR, the spin of nuclei in the magnetic field. Spin. So usually you have here for the hydrogen and the carbon NMR. Now, if you're going to see how light interact with matter, usually if this is the matter and light comes in, these are the possible modes that happen. Light can be transmitted, reflected, absorbed, or in some case, they luminous, like what fluorescence is, or they scatter the light. So based on the different light matter interaction, there's a corresponding method that was developed. Okay, so the idea to make it more into a quantitative analysis is to put some measurable units in it. Okay, because spectroscopy itself is just, we could say, the interaction of light with matter. But if you put some sort of a quantitative relationship on how much was absorbed, how much was reflect, how much was scattering, you're doing there some sort of a spectrophotometry. Okay. So the main experience that I have is absorption, luminescence, and scattering. Okay, scattering is the Raman that I have worked on. Absorption is just the UVBs. Luminescence, there are two types of luminescence. One is the so-called static, and the other one is dynamic. So when we're talking about static, when you undergo fluorescence, so what happened when light hit the molecule, it is excited. So when it goes to the excited state, if it's a fluorescent sample, okay, before it goes back to the ground state, it will pass through a transition state. Okay, and when it passed through the transition state, sometimes they last there for a certain lifetime. So if you're looking at the steady state, that's just emission, excitation and emission. But if you're looking at the dynamic state, it has something to do with what we call the lifetime. So both of them are what we call a very good technique. Okay, especially if you're looking at the biological nature or interaction in biological setting. So when we look at the molecular spectroscopy, it's used for the identification and determination of a huge number of organic, inorganic, and biochemical species. I think after the balance, the next thing that you need is the next instrument that you need, okay, would be a spectronic 20. With a spectronic 20, you can do a lot, okay? Because if you have the ability to convert a target analyte into a colored reagent, and the intensity or the absorbance of the colored reagent can be measured, then you can develop already there a quantitative method. Okay, so if we're going to look at the molecular ultraviolet and visible absorption and fluorescence, they employed primarily for quantitative analysis. You seldom use IR and Raman for quantitative analysis. You use it for structure elucidation. Okay, usually where do we use IR? Anyone? Saan tayo nag-IR? To those who took their undergrad in UPLB, where do you encounter IR? Anyone?
Identification of functional groups.
Okay, so IR is mostly on what we call functional group. The same thing with the Raman. Okay, there's only what we call the difference between the two, okay, on the samples that they are what we could analyze. But both of them are used for functional group determination. Okay, and in addition to that, both IR and Raman can be useful to tell you the environment of a given molecule that you are analyzing. Okay, and I'm going to show you some examples that I have through the years. So as I told you, this is a very common technique. Okay, I think in the industry, all labs would have this one, the use of the so-called scattered light. So it's applied in chemistry, pharmacy, cosmetic, food, and agriculture. Okay. So what I'm going to do is the different, we could say, experiences that I have using spectroscopic methods. So you're familiar with this paper wherein we analyze the effect of tetracycline antibiotics in pinto beans. So aside from the mass spec, we also use fluorescence during this time. So what we have observed is if you have chlortetracycline, okay, it can become fluorescence once it forms a complex with calcium. And it has been there has been a saying that if you're going to take tetracycline as your antibiotics, you have to be very careful, because one of the side effects of having tetracycline in your body is it will give you a glowing bone or teeth. Okay, because calcium tetracycline complex are what we call the fluorescent material. So what we observe here, when we treat our plant with tetracycline, we have a higher intensity of the emission scan. So we excited around 360, okay? And then we found out that only the treated plants are excited, but the other one, the untreated one, because there's no tetracycline in it, okay, it's not what we call, its intensity is lower compared to the one that is treated with tetracycline. And the explanation there is the calcium, the intracellular calcium that we have in the plants, okay, form some sort of what we call a complex with tetracycline, and this results to the stagnant, or we could say small leaves, okay, for those treated with tetracycline, because what happened, the available calcium that should be used in other processes are not available anymore. It binds with the tetracycline. And then I also used absorbance, okay? So we're trying to determine, so I'm trying to develop materials that is used for solid phase extraction of tetracycline. So what we found out, that tetracycline, which is usually affected by the environment, so the one that is being imprinted in our zero gels is not really tetracycline. So what I did is I tried to monitor them. So the first thing that I did is using absorbance in determining the washing, because the way that we do when we prepare the zero gels, this is like a glass-like materials. We put a lot of tetracyclines in it. And then what we do, we remove the tetracycline by solvent extraction, okay, hoping that there are some imprints of tetracycline that we can use, okay, for complex samples like soils and water that contains tetracycline. But what we found out, that the tetracycline itself is changing when it is being imprinted. So from the initial days that we have here, okay, after several days, you could see the peak around 360, okay, shifted, which means that another substance is forming, and we were able to identify it using mass spec or chromatography, which I presented to you during our what we call chromatographic experience. And then we try to mimic it. What if we put it with an acid? Why we put it on acid? Because whenever the sol gel hydrolyzes, it releases in it some acid. So we try to mimic the condition, okay, inside the zero gel, and we found out that there's what we call the formation of anhydrous tetracycline while tetracycline is being imprinted in the zero gel. So the first thing that we use is absorbance, and then the same sample that we have here for absorbance, we use it to analyze what compounds it has using mass spec. And then we also try to use, let's say, the PLE washing. So this is a pressure liquid end cup, pressure liquid extraction washing. So we use what we call tetracycline with different recipe, and another tetracycline, which is what we call OTC. And we found out that we lose the peak around 360, because that's a signature peak of tetracycline. So when I moved to Pace University, these are the spectroscopic instrument that we have. So I'm not sure if you're familiar with this one. Can you tell me what is this, the one I'm pointing right now? What do you think is this one? So this is an IR, and this is a spectropolarimeter, this is a UV-based spectrophotometer, and this is a portable Raman, and this is a microplate reader that can do absorbance and fluorescence. So that's the one that I have at Pace University as the instrument that is, we could say, spectroscopic in nature. Okay, and the projects that I usually have is we use instrument, okay, to analyze compounds that are of interest. So one of the instruments that we have there are the spectroscopic base instrument. So usually we analyze the sol gel, the proteins, okay, the pharmaceuticals and the antibiotics, and mostly these so-called natural products. But in my other lab, which is in your college, the instruments that we have here is, we could say, more high-end compared to the one that we have at Pace University. So if we want to get data that is worth for publication, I usually validate most of the results that we have from Pace at your college. So we have UV-based, okay, spectropolarimeter. This is much better than the one that I have at Pace University. And then we have IR, okay, and this one is the benchtop Raman spectrometer, which is much better compared to the portable Raman spectrometer. And in addition to that, we have this so-called circular dichroism spectrometer, CD. So CD is used for confirmation if you're studying biological samples. Yung tinatawag na alpha helix, beta sheets, yan yung ginagamit na instrument. Okay? In fact, they have two CDs in your college. The problem in your college is we don't have enough personnel there. Okay? And whenever I go there, usually 2 or 3 instruments are running simultaneously. All I need to do is when I have the cuvette, from one instrument to another, except the IR. Okay? So one of the studies that we did there is looking at the interaction of this so-called amyloidogenic protein. So what we're trying to do is, why does some protein fold? Okay. And protein folding is associated to the so-called degenerative diseases like Parkinson's, Alzheimer's, and even diabetes. So what we look is one peptide, okay, one protein that is involved in this diabetes, and it's called amylin. So what we look is, we look at a certain fragment in the amylin. So the one from, we could say, 22 to 29, okay, the peptide there. And we synthesize, okay, this peptide in the lab. And what we do is we just change the functional group that is attached to phenylalanine. Okay, so what we do, the R group that we have here, especially the R1, we change it with different functional group. Okay, and we see if the peptide that we have will be what we call aggregate. When I say aggregate, form solid or form an amyloid thing, or it will remain, we could say, in liquid form. Because the way that we're trying to do is how does this ring affect the amyloid formation? So we're trying to look at the environment. So what happened if there is amyloid formation? What happened when there is a solid aggregation of the peptide that we have? So we monitor them using different spectroscopic method. Okay, so we do the turbidity study. So this is using absorbance at a certain wavelength. So usually what we observe, if the peptide aggregate, there's an increase in the absorbance at 405 nanometer. Okay, and then after it aggregates, it will what we call, when all the solids go down or settle at the bottom, the absorbance decreases. So we're able to find some of these library that we have, some of them showed high absorbance. One of them, I think this is the cyano, it takes a while for it to be aggregated, okay? And at the same time that it's aggregated, we also try to monitor the fluorescence, because the native fluorescence of this peptide forming these solid aggregates also give us fluorescence or high intensity in terms of fluorescence. So if we're going to match here what we get in the absorbance and the fluorescence in the main parent of the polypeptide that we have, so they really match, okay, in terms of what we call time. So this is a time dependent analysis. And then in addition to that, if we want to get more information about what happened, okay, to the peptide that we have, we use the vibrational spectroscopy. And this one, the IR, since we found this signature peak at 1628 wave number, so this is a signature peak for those formation of beta sheet. Okay, so whenever there's amyloid formation, usually most of the materials that they have there form beta sheet conformation. And we try to compare the peptide, the aggregate before and after aggregation. So as you could see here, after aggregation, there's a peak around this region, which is around 1675. This is using Raman, okay? And if you're going to look at the peak at 1600 and the peak between 1300 and 1400, they usually represent a certain functional group. Now, since this is a nitro, okay, so this is one wherein the functional group that we have here is NO2. So nitro has, we could say, a sharp peak at this region. And if you're going to look at the peak at this region before aggregation and after aggregation, there's some sort of a slight shift in the peak, which means there's a change in the environment, okay, of the functional group. And the 1600 that we have here, that represents the ring modes. And when they aggregate, there's also a slight shift of the peak. But the one that is really distinct here is the peak around this one. So as you could see, before aggregation, because they're in a liquid form, you don't see any peak at this one. But once the peptide aggregate, you see a peak here. So that is what we call a beta sheet mode. So whenever you have a peak around 1670 to 1680, it means that there's the formation of the beta sheet, which is, we could say, a signature of amyloid formation. Okay, so if we're going to look at this, to look at the change in the number, so this is the same data as this one. We just swap the numbering here. So from 1356, when it aggregates, it becomes 1351. From 1608, when it aggregates, it becomes 1605. So the so-called downshift here means that your environment becomes more nonpolar. Okay, if the shift is on the other side, then it becomes more polar. Now this one here, it just shows you the 1674. It just shows you the formation of this beta sheet. Now we also follow the slow forming peptide, the cyano one. So what happened here before aggregation, this is what we have. And then when the aggregation starts, I don't know if you can see the red one there. Okay, but if you're going to look at here, sorry for the inconsistency of the shift here, there's really a downshift there, similar to what happened in the functional group and the ring mode of the nitropeptide. And then once there's what we call complete aggregation, okay, it becomes more, we could say, downshift. I mean, the wave number that you have there is much lower. Okay, and we follow this one. This is the one coming from our publication. So we start at 2 to 51, and then one starts to what we call aggregate is 2239, and then with aggregate is 2234. What happened is, you might say, why is the signal increasing? What do you think is the reason why the signal is increasing? Anyone? Bakit tumataas yung signal niya or yung peak? Because remember, from a liquid, it becomes solid. Okay, so when you say aggregation, you form some amyloid there that is usually solid in form. So they become more concentrated. So tumataas yung signal niya. Okay, now part of the things that I did also in grad school that I asked my student to repeat at Pace University is we look at different pH of this tetracycline. Since we noticed that at different pH they change, so we try to monitor them. We use what we call different analyte. So this is just, we could say, the absorbance that we have here. So as you could see here, okay, as the pH changes, this is metacycline, minocycline, rolite tetracycline, and oxytetracycline. But I have around 10 tetracycline analogs that I have there. But I'm just going to show you this one. So at different pH is changing. And we also observe that when we look at the fluorescence, it's also changing. Now, in addition to that, we also have the lifetime. So to those that are, we could say, most fluorescent, like your chlorotetracyclines, which at higher pH becomes isochlorotetracycline, we're able to see the difference in the lifetime, okay, when they change in their pH. And then we still have this what we call Raman thing. So what we did is we look at this cyanohydroxycinnamic acid because we are curious with the isomers that we have. Why is it the 4-cyanohydroxycinnamic acid can be used as MALDI material, but the 3-cyanohydroxycinnamic acid is not possible? So we compare it with the hydrocinamic acid and one that doesn't have the what we call cyanocinamic acid. So this is, we could say, the hydrocinamic acid. Okay, and then we have here the cyano. So if you're going to look at here, the difference that you have here is just this. So this is, we could say, the 3, and this is the 4, and this is the one that doesn't have the hydroxy, just cyanocinamic acid. And when we compare the peak, so we found out at the wave number around 3300, only the 4-cyanohydroxycinnamic acid has a peak. So what does this region represent? Anyone? Recall your Orgo or 140? What does the 3300 represent? What functional group do they represent? Wala? Nakalimutan nyo na?
Group.
Ha?
OH group.
OH group. So if you're going to look at the functional group that we have here, doon sa structure, so may OH dito, may OH dito, may OH dito. How come the OH of this is more prominent compared to the one in this position? Okay, so that's the one that caught our attention. So the initial thing that we do is we just compare the Raman at different region. And then we also look at this region. Now this region is what? Anong functional group yung nasa region na to? Anyone? This is what? This is the CH region. Okay? And then we also look at this, what we call 2200 to 2300. And as you could see, only the hydroxycinnamic acid is not what we call the showing or exhibiting any peak, because this peak is the peak due to the functional group cyano. Okay, so we just do it like that and we're able to get the paper. And then there's a follow-up on that. Okay, so this one we already described why the 4-cyanohydroxycinnamic acid is a good MALDI material compared to its isomer, which is the 3-cyanohydroxy acid. Although Raman is the main we could say instrument that we use, we also use NMR. And then we also did some sort of quantitative determination of estrogens using Raman. And in one book that was published by the American Chemical Society, the Zesamero group and my group was able to publish 5 chapters there, of which 4 I'm included. Okay, so the first that we have there is like, how do we integrate Raman spectroscopy in undergraduate instruction and research at my institute, at my home base. And then we have is still the cyano. Okay, so solvent sensitivity of the cyano group, a Raman spectroscopic study. So we just look at the different compounds that contain cyano and try to look what will be the effect if, let's say, we change the polarity of the solvent, we change the pH. So we just what we call do stop, and we have an experimental result, and at the same time we also have theoretical results. Okay, now one thing that make us, we could say, interested in the Raman spectroscopy, we have a forensic science program at Pace University. So we use even that what we call portable Raman. Okay, the students are trained to what we call use that thing. And what do you think is the material that they're going to use? So usually, okay, we test them using any nitro-containing compound. Why nitro? Anyone? What's the relationship of nitro, let's say, in forensic science? Anyone? Bakit nitro yung ginamit namin? Nitro is a what we call sensitive functional group. But why do you think nitro? Active ingredients usually ang nitro ng ano?
Explosives.
Explosive. Okay? So parang ang ginagawa namin is we training them to detect MAC, explosive. Okay? So yan yung isang thing na tuwang-tuwa yung mga undergrad namin because they feel like they're doing real stuff. Okay? But what we put there is just different nitro compound. And then another student of mine, okay? I'm not sure if you're familiar with the silenes. So usually there are 3 isomers of silenes. So what we did is if we can do some quantitative analysis using silene mixtures. And we use the portable Raman. So what we have using the portable Raman, so we just have P-silene, O-silene, and M-silene. And what we did here is we found we used some sort of a common peak. Okay, and we get the ratio of the common peak with a unique peak of each of the isomer. So we test the mixture O and P-silene, M and P-silene, and then M and O-silene, and then MO plus P-silene, and we try to develop a method, okay, that should be able to quantify silene mixture. And then the very popular one that we use, okay, this is the so-called over-the-counter drugs. So I think you're familiar with what we call acetylsalicylic acid. Okay, acetylsalicylic acid, that's the AI4. Anyone? Saan siya active ingredient? That's the active ingredient of aspirin. And then you have acetaminophen, which is known in the Philippines as paracetamol. Are you familiar with that? Okay. Acetaminophen is just paracetamol. And now about ibuprofen. And then we have naproxen. And where is the guaifenesin active ingredient of? Anyone? So these are all over-the-counter drugs. So these are you can buy without prescription. Okay. So most of them, I think naproxen and ibuprofen and acetaminophen belongs to this NSAID, non-steroidal compound. So guaifenesin is just doon sa tinatawag nating cup syrup. Okay. So what we did, using the portable Raman, we try to find unique peaks. So as you could see here, the signal to noise is not really good if you compare it with the benchtop Raman. So what we try to do here is we try to see if we can differentiate, look for the unique peak, and if we're given, okay, a powder, all we need to do is compare the Raman spectrum of that powder with the different known active ingredient of this, what we call over-the-counter drugs. So this is the common name that they have. So Aleve is your naproxen, Tylenol is your acetaminophen, Mucinex is the guaifenesin, okay, and then we have a generic ibuprofen, okay, and then we have Excedrin, which contain aspirin and acetaminophen, and then we have the aspirin. Now, most of the thing that we do in the lab is what we call in Propolis. Some of them, their samples even came from the Philippines. So we have here, I don't know if you could see here, there's what we call Bicol, UPLB, and Tayabas. And then this is from other countries. So all I can say here is Australia, Latvia, that's CA, that's California. But if you're going to look at the Propolis, usually they are different colors. Why? Because their composition depends on their locations. It not only depends on the location, it also depends on the what we call weather or season that they have. So it's a very good material to be analyzed. It's hard to characterize them because they will change, their composition vary. Okay. So what we did, the one that I got from the Philippines, so we compare the absorbance. So as you could see here, okay, the one from Albay, Quezon, and Laguna, they exhibit different absorbance peaks. But it is within, we could say 250 to 350, which is signatures for the so-called phenolic compounds. And what I did, I tried to differentiate it using fluorescence. So I excited them here at the wavelength that is around, if I'm not mistaken, 320. Okay, because we're trying to get here the maxima. So whatever the maximum peak in your absorbance, that's the excitation wavelength that you're going to use for fluorescent. And then we also use IR. So as you could see it, if you look at the naked eye, it seems like they're not different. It seems like they're the same. So how are we going to differentiate them? Okay, so there is a process called derivative spectroscopy. So what it do is you make, right now, all the data that you have here are zero derivative. So you can make a first derivative or a second derivative of those raw data, and you could see the difference. Another way that you're going to do it is what you're going to do next week is using principal component analysis. And you can see there that each of them are different from one another. Okay, so another paper that we have here, we use different extraction methods. And based on the absorbance, okay, the ultrasonication is the best one, followed by microwave. Okay, and then we also use fluorescent. And in addition to that, we also use GCMS last time. So right now, okay, so this is a deep, if I'm not mistaken, this is around 290, because you have a peak here at 290, and then we excite them also at this peak around 340. Okay. So at 340 is the microwave, the one that has the highest, what we call the emission scan compared to, which is also this one. So what does this mean? Microwave is not as high here. So it means it has more fluorescent materials compared to the other. Sorry for the noise. That's what we get living here in 3rd Avenue. Now, the thing that we use right now, we try to what we call use FTIR to study microplastic. And the sample that we have, believe it or not, are coming from this turtle poop provided to us by a biology professor from another university. So we try to look at the signature that the plastic has, and we try to see if we can get it from the poop. And then we use the microplate leader to look at the antioxidant activity of T and B products, including propolis. So these are just the assay that we have. So this is a DPPH assay. This is ABTS assay. So these are antioxidant-based assay. And this is the Folin Shulka 2, which is used for phenolic contents, because it's really what we call colorful. Okay, almost every week we get some results from the students using this one, and we use our microplate reader. Okay, now the experiment that you're going to have is based on what we call the AAS. And what you have there, I'm going to send to you maybe later this week. I just want you to concentrate on the exam. Okay, our data. So if I'm not mistaken, how many groups? There's 3 data from these 3, we could say, serials. And what you're going to do is you analyze the iron content. Okay, you're familiar with iron, right? I don't know if you're going to believe me, but what we do here in one of the experiment, we have these cereals, and okay, we ano ba tawag dito? We pulverize it, okay, and then we have a magnet, mix it for 1 hour, and after 1 hour, we could see some gray stuff sticking to the magnet. And what we do, we remove that gray stuff by mixing it again for 1 hour, okay, in a heated HCl. Now, you know what that gray material is? That's your iron metal, okay? And then we use the AAS, okay? Prefer the calibration curve and then get the reading, and then compare if the amount of the iron that they have is the one that is shown in their label. So usually the experiments that we have here has something to do with consumer products. Okay, remember your HPLC? So we also use their consumer products, which is what we call the soda. So the same thing that we do in the other experiments that we have here, so that the students will be interested that, oh, all the stuff that we do is related to their everyday life. So I don't know if you saw the video that I posted, that was yesterday. So I have a class at your college. It's an instrumental analysis also, but for non-science major. So what we're trying to work on it yesterday is the AAS that we have. So for 30 minutes, I have a hard time turning it on, because if you look at the video, the flame doesn't reach the burner. So what I did in the end is the same thing that we did in our what we call chalane or gasol stove there. So I get a piece of paper, light up the flame, and then put it in the burner. That's why we end up getting the reading. So the experiment that we did yesterday is determining manganese. This time in steel samples. Yung ginagamit sa mga construction. Okay? So usually if you have AAS, it is a single element method. Okay? So based on atomic absorption. So the more atoms that you have there, the higher is the signal that you have. So this end my presentation here for today. So may question ba? Tanong? May tanong kayo? So sino dito may experience na sa absorbance? I-ano nyo yung raise your hand function or approve. Either yung ganun or yung ganun. Sino may experience sa UVBs? Wala? Wala? Hindi kayo nakapag-UVBs sa buhay nyo? So almost all. Okay? Sa fluorescent. Lahat kayo na ano sa fluorescent? The classic, I think, experiment of fluorescence is, I'm not sure if you know, quinine that was used for to treat malaria. So usually if you have this tonic water, okay, that's the experiment that we do. So we determine the quinine content doon sa tonic water. Okay. IR, you have any experience with IR? Anyone? Very good. Nagpa-function ba IR natin sa IC? Buhay pa ba yun? Because I remember using, oh no, I was not able to use it. It was new during that time. And I had to go to the forest product FPRDI to have my sample analyzed for IR. I'm not sure if any one of you has the Raman, what we call experience on Raman. How about AAS? Naka-experience na kayong gumamit ng AAS? Usually AAS is for what? Metal content. Okay? Question? May tanong? Ready na kayo sa exam? And also, I'm not sure if you're familiar where to download your MATLAB. Usually, yung MATLAB na yun ay, I think it's, pupunta kayo sa link na kagaya nito. MathWorks. And you can only obtain that using your UP account. I was able to obtain it not because of my PACE account, but because of my other school account, my what we call CUNY account. And it's going to look like I'm not sure. I try to open the MATLAB that I have. So it's going to look like this. So I'm opening my MATLAB. Once it's open, I'm going to share it to you. So I don't know if you can see it. Sorry for the noise from my daughter. Wala kasi silang klase bukas kaya gising pa. But I have my class. It takes a while to open. So share ko ulit. So this is how it's going to look like. And I've been trying to what we call play with it, okay? But Dr. Carl Pilario will discuss about this next week. Although I'm going to give you some sort of I'm trying to share the video that we have here. Share sound, optimize video clip, advanced. Desktop, trying to show.
Yan, hello po.
Do you see
So
the
what we
video?
have here is the data set. And yung columns yung ating mga samples. And
Kita nyo
yung
ba?
rows ay yung ating mga features. So, ang ginawa ko po muna dito sa kanan, yan, naglagay lang po ako ng classes. So, for example, class 1, Washington, na hindi sonicated, then Washington sonicated is class 2, class 3, then 4, Bulgaria, 5, England. So, lahat po ng headings ay nilagay ko dito sa listahan na to na 54 pala sila lahat. And we have a total of 13 classes. So, una ko pong gagawin is from here, I select all the data. Haba pala. Okay, then copy. Tapos, I have MATLAB open already. Okay, then dito, I'll create a right-click sa workspace. Then, create a new variable, Propolis ATR,
Pa?
for example. Then, double-click that new variable. Then, paste the data. So, purely numbers lang yung nandito po. Yeah. Okay, 3,401 and 54. Correct. Now, for the purpose of the analysis, kailangan baliktad, which means dapat yung columns pala yung mga features and yung rows yung mga samples. So, para matranspose siya, lalagyan ko lang ng Propolis ATR is equal to Propolis ATR transpose, semicolon. So, yun, nagbaliktad na po siya. So, now we have the number of samples dito sa rows. So, hanggang 54 na lang yung rows. And then, yung number of columns, yun yung hanggang sa 3,401. Okay, in addition sa Propolis ATR na data po, ay kailangan din nating i-transfer yung information ng classes. So, right-click again. Let's create a new variable called class or classes. And then, double-click. Itong information na to for the 54 samples ang ilalagay ko rito. So, now, ito na po sila ngayon, 54 elements. And then, pagdating po sa code, so, pupunta po ako sa editor. Yan. I also need to put the class name. Class name. So, ito po yung listahan ng unique class names. Yan. Tapos, kailangan po ang pagkakasulat ay naka single quotes para ma-read niya as string. Okay. Tapos, nalagyan ko lang ng comma to separate the different strings. Quezon And last, close. Alright, so now we have all the information, class names, class, and the actual signals ng Propolis. And then from here, pwede na mag-PCA. So, I'll type coef score equals PCA of the Propolis ATR. Yeah, so ibig pong sabihin ay this code will already perform principal components analysis. And then, yung score, yun po yung kailangan natin. The first two columns of score are the first two principal components. So all we need to do is to create a plot or a scatter plot. In this case, let's try G scatter and then score of the first column and score of the second column. Yan, dito po papasok yung classes or yung class. Kasi i-read niya yan as the parang gagawin niyang same color lahat ng same number sa class. And then, lagyan na po natin ang legend na for each of the class ay class name yung legend. Okay? So, let's save this as Propolis PCA. Actually, any name will do. Then, let's try to run. So, ito na po yung result. Okay, lalagyan ko lang po ng grid. So, type ko lang grid on para magkaroon ng grid. Ayan. So, now, ayan. So, ito po yung mga lahat ng class names. And ito yung pagkaka-arrange nilang lahat in this plot. So, kung titingnan po natin, yung Washington ay yung red. Okay. Washington sonicated yung orange. So, medyo malayo. Ang magkakalapit ay itong Greece sonicated. Malapit sila. Whether or not sonicated pala. Okay, so yun lang po.
Okay, yep, I'm going to what we call post it. Okay, para makita nyo. But as you can see, ano sa tingin nyo? Kaya ba? The good thing there is Dr. Pilayo doesn't have any idea doon sa data na binigay ko sa kanya. All I said is, I want to see if there's a difference because akala niya dapat magkapareho, but they are the same sample, but one was just extracted, the typical soaking method, while the other one was sonicated. So kita mo doon, magkaiba yung reading. He used an ATR, which is a component similar to IR. So, tama yung results na dapat magkaiba sila. Because we're trying to see if there's a difference if we sonicate the Propolis sample compared with just soaking it, the typical maceration or soaking method. So, what you're going to do is you'll be given absorbance or FTIR data na galing sa mga students ko. And Ma'am Ivy and I decide if you want that to be your what we call special topic, we can talk about it. So try to what we could think about it. Because if you can get something out of it, like maganda yung pagkasulat nyo, maybe we can submit it for publication. But the idea there is at least malagay sa resume nyo na meron kayong experience mag-MATLAB. Because I think that's the best thing that we can get out of this online mode that we have. Wala man kayong experimental experience, but at least you're able to do MATLAB. Because the trend right now is the experimental methods are not enough. You have to do or use the so-called theoretical and sometimes chemometric method. So ang bala ko nga gawin, yung mga gagamitin nyo, yung mga raw data na ng mga published papers namin. And see if they're really what we call difference. That's the safe way to do it. So tanong, Ma'am Ivy? May tanong? Kita nyo, I mean, Stephanie and I think Emil was in Paase last night, and you see how they presented their data there. So yung iba nag-ano sila, nag-principal component analysis doon sa data nila. I think the one with the, is that the dieting from Bart? Okay.
Yes, sir.
So as you could see right now, that's really a very what we call useful method. Kasi minsan absorbance lang, tingin mo lang difference, but sometimes what you see in the naked eye hindi talaga I mean kita. So you try to process the data that you have, okay, and use any software. There are we could say free software. I'm not sure if you're familiar with the software R. Did you hear anything about R? Narinig niyo na ba yun? May experience ka doon, Anthony? Sa R method or sa R software? Because a friend of mine here at Pace University, she's a biology, biostatistician. Yun yung ginagamit niya. Kasi daw libre. But since Dr. Pilario is a chemical engineer, he's a faculty in Department of Chemical Engineering in UP Diliman. So they said if it's engineering, they use MATLAB. And he's the one who informed me that, oh, it's UP system has access to that. Okay. So why don't you ask your student to download it? Kasi daw libre. That is a student. Kasi ginagamit ng mga students niya. So question. May tanong, huwag mahiya. Alas 10 pa lang. I'm trying to see if I can download the video I sent you. Because what happened, I'm trying to upload it in my computer. Eh, ang bagal. So sabi ko, ano ko na lang kaya to? So I just try to show you here how we operate this thing. Medyo. Show screen. Basic. So what this is when we're trying to light it up. Oh, that's the one. Ito yung initial attempt ko. As you could see there, the flame doesn't reach the burner. So, ano dapat dun? But it's dun, wala akong matchstick. So, ang ginawa ko, kumuha ako ng papel, yun, nag-work din siya. So, medyo magulo lang kasi ako yung nagre-record dyan eh. So, I don't know if you could see how the flame become more intense when you put the sample. So that's the experience that we have in AAS yesterday. We're doing in-person class. So you see only, I think, here how many voices there because most of the class is on the other room. The instrument is on separate room. So question. Tanong.
Good evening, sir. I have a question po. Pero
Yes,
not
Jeff.
so much about Spectro po, sir. About the exam, sir. Ano po ba yung magiging scheme? Will it be timed or open notes? Ganyan po ba?
Ibibigay kayo ng question. It will be posted by Ma'am IB. Tapos sasagutin nyo yung question na yun. You will be given time. Pero hindi naman siguro one day. I mean, there's a time frame that you need to what we call finish it and then upload it.
Okay po. Thank you, sir.
So it's a set of questions. Walang calculation. That's all I can say. It's more on getting to know how you understand the concept of the instrumentation, especially yung first part natin, chromatography. So parang, kung gusto mo mag-analyze itong sample, so ano yung gagamitin mo? GC or LC? Tapos kung GC or LC, ano yung detector? So ganun parang tanong.
Okay, sir. Thank you very much po.
Yung iba, mukhang matindi meeting ni Ma'am IB, hindi siya makasagot. May tanong pa? So, please, before tayo mag-meet next week, I suggest you download the MATLAB already. Because what we're trying to do is the data that Dr. Pilario will demonstrate to you will also be made available to you. So parang nagdi-demo siya doon, ginagawa nyo rin yung same set of data. So this is in addition to some short lecture that he will discuss. PCA is only one we could say calculation, but you can use the MATLAB in other analysis. Maybe it will be useful to you in your thesis or dissertation. Kasi may access na kayo. Anyway, that's an academic, what we call access. So you can install it in your personal laptop. May tanong? May tanong pa?
Hi,
Okay,
sir.
Stephanie. Yes.
Ano po, yung deliverable for this week po is yung sa exam lang po.
Exam.
Sa excerpt po, wala.
Wala pa. Yung AAS ipapadala pa lang namin this week sa inyo. Pero I think
Okay.
you're usually given what? At least 2 weeks kung magsulat kayo ng report about nun.
Okay po, sir.
It's
Thank you po.
It's a pretty straightforward experiment. You have a calibration curve, data, tapos yung readings ng mga sample nyo. So parang ano lang din yun sa caffeine experiment. So ang
Thank you po, sir.
sige, yung deliverable sa inyo this week is just the exam. So may tanong pa? Yeah, I think pwede ata mag-stat analysis sa MATLAB. It has what we call a lot of analysis that you can do there. Kasi usually ginagamit yan sa AI eh. Yung specialty kasi ni Professor Pilario, I'm not sure if you're familiar with machine learning. So, pwede rin ata siyang gamitin sa artificial neural network. Yung i-train mo, tapos once ma-train mo, pwede mo siyang panggamit, pa-detect kung ano man yung sample na train mo at i-identify niya yun. So I would say take advantage of that software. The earlier that you can download, the better. So at least that's the best thing that we could leave to you, especially at this time of pandemic. Good thing you have it. The previous semester, they don't have it. If you have friends who've taken the course last time, they don't have that software. So yun yung parang advantage nyo kaysa sa kanila. Question? Anthony? Amiel? Kim? Karen? Jeb? Stephanie? Herian? May tanong? So with that, abangan nyo na lang yung message kay Ma'am IB. I already saw the exam and I approved it already to be given to you. So once I think we're done, hopefully she can post it. So you can utilize the time that we have today to start solving the exam. So kung wala nang tanong, hey, see you next week. Okay?
Thank you, sir.
Bye po.
Bye.
Yeah, po, sir.
Thank you, sir.